we remember anything from last class? Do you remember what was the Mendel's law? No? Well, Mendel, we talked about two laws of Mendel, right? What was chapter, it's 11. I'll just go briefly on chapter 11 and then go over again with chapter 12, okay? Chapter 11, Mendel is called the father of genetics, right? His first law is law of what? Law of segregation. What do you mean by segregation? Right, there are some two entities that determine our outcome, the phenotype, the genotype, right? It always comes with a pair, okay? The two things is separating when we make our gamete, when we make sperm or egg for the next generation. That's the segregation, okay? What we call it alleles, right? We can have two alleles, but within population we can have many different alleles of the gene, okay? That was the first law. Second law was law of independent assortment, okay? Independent assortment. In this case, we have to think about two traits at the same time. For example, yellow versus green, the color of peas, or flower color, or wrinkles, or round, two characters at the same time, okay? So independent assort means, if you think about meiosis, okay? Meiosis. In meiosis 1, what happened? Two homologous chromosomes are together, right? There's some exchange of chromosome. But at the same time, the question is whether they can line up this way or this way. That will determine which way one of the homologous chromosome will go, right? So if you think about chromosome number 1 and number 2, each case is independent of each other, okay? So it's independent assortment of chromosome, which will turn out whether, look at the color of the flower or color of the seed, it's independent of each other, okay? That's the second law, okay? So now, there was some exceptions to the Mendel's law, right? What was the first one? It's called incomplete dominance. Okay, if you mix between white cauliflower and red, the F1 comes out as a pink, right? It's not mixing. It has two alleles, but one is not completely dominant over the other. So it's called incomplete dominance. Then there is called codominance. What is the example of codominance? Blood types, A, B, O. Okay, you can have A and B, and your blood type is A, B. Neither is dominant nor recessive. Okay, then there's a very complex traits, phenotypes, like our height or the complexity of our skin. It's not, yellow or green, or it's a between. There's so many different traits. Okay, those are determined by many genes, not just one gene. Okay, that's called polygenic. There are many genes that control the traits. Your heights, So in this case, the characteristic is it has a variation of the phenotype. Okay. What else? What else was there? That's about it, right? So we're moving to chapter 12. Okay. The units of heredity. Okay. So now we know about chromosomes, right? And then we talked about X-linked inheritance in humans. Okay. Certain human conditions, such as red, green, color blindness. Okay, there's the three color, red, green, and what is it, blue? Yeah, but particularly red and green, this person cannot tell the difference. Okay, it's called red-green color blindness. The second one is called hemophilia. Okay, having problem of blood clotting. So if you have big cut, the bleeding keep bleeding. So sometimes it can be fatal. Okay, those two genetic disease, the observation is somehow males showing more frequently of this disease. So they didn't understand. But now we understand because the gene that affects that disease is located on X chromosome. Okay, X chromosome. Okay, so that's why we called X-linked inheritance. So if you can read this number 8, you're okay. If you cannot read, you're, red-green color blindness. Okay. So men are more likely than women to suffer of these conditions because men have only one X chromosome and then one Y. On the other hand, female has 2 X chromosomes. Okay. So even if the female has one affected X chromosome, they can have normal X chromosome, which can help. Okay. So women with dysfunctional blood clotting allele, which is hemophilia allele on her X chromosome, usually is protected from hemophilia by the other X is functional. Okay. And we can skip. So this is how we can understand the X-linked inheritance. Okay. So in this case, female mother can have 2 X chromosomes. One of them is affected, but she is not color blindness or hemophilia. But she married to, let's say, a normal male, and their children, okay, can have this combination. She can have 2 X chromosomes separated. It's a law of segregation, right? And the male gamete X and Y. So next generation, this is called Punnett square. Next generation, this sperm will meet this egg, and it's normal. And this X would meet this X, 2X. She will be a carrier. She can carry this affected allele, but she doesn't show that condition. Okay? But in male case, if you have only one X chromosome that is affected, that will show up. Okay? That's why male show higher frequency of these conditions. Okay? Understood? Yeah. Not difficult, right? So we call this person who carries the X chromosome, affected X chromosome, but that does not show, we call her a carriers. Okay?
Various human genetic disorders are said to be X-linked, which means that the defective allele responsible for the condition is carried on the X chromosome. This implies particular patterns of inheritance because X is a sex chromosome, with females having 2, but males only 1 copy of X. As a consequence, X-linked diseases claim more male than female victims. This imbalance is explained by the animation on the left, which illustrates inheritance of an X-linked disorder, red-green color blindness. This is an example of an X-linked recessive disorder because the color blindness is not expressed if a single functioning allele is present. That is, if we denote the normal allele by uppercase X and the defective allele by lowercase x, the genotype must be either lowercase x, female, or lowercase x, uppercase Y, male, to express color blindness. However, a female of genotype uppercase X, lowercase x, is a carrier for the disorder. Although she will not express the recessive trait, she can pass the defective lowercase x allele to her offspring. As the animation illustrates, the chromosomal makeup of males makes them more susceptible to X-linked recessive disorders than females. For a daughter to express the color blindness, she must inherit 2 lowercase x alleles, genotype lowercase x, lowercase x. But a son who inherits a single lowercase x allele will be colorblind, genotype lowercase x, uppercase Y. As a consequence, color blindness is much more common in males than in females.
Okay, that's why we call it X-linked inheritance. Okay, so that's about, the gene is located on X chromosome. X and Y, we call it sex chromosome, right? So it's called sex-linked disorders. Now we're going back to autosomal. In other words, chromosome number 1 to number 22, genetic disorder. In autosomal genetic disorder, you have to understand whether this is dominant genetic disorder or whether this is a recessive one. Okay. So sickle cell anemia has many names, Korean names, right? It's an example of autosomal recessive disorder. Recessive disorder means you have to have 2 copies of it to show this disease, okay? Because by definition, recessive means if you have as a heterozygote, if you have one normal, one affected one, you're supposed not show it. That's the definition of recessive, right? So you have to have 2 copies to show a disease, okay? This sickle cell anemia, their red blood cell looks like this, okay? This is normal donut-shaped red blood cells, okay? And if you have sickle cell anemia, they'll produce red blood cell shaped this way. And they don't carry enough oxygen to your bloodstream. That's why you're having anemia, okay? But this disease somehow, the heterozygote, right, somehow this disease is a little bit resistant to malaria. That's why sometimes, this affected allele is persistent in certain populations. Okay, we'll come back to that point in later chapters. But first one is, this is autosomal recessive genetic disorder. Okay, it is recessive because person must homozygous for the sickle cell allele to suffer from condition. Homozygous means you have to have the same 2 copies. Okay, so they must have 2 alleles that code for the same sickle cell hemoglobin protein. So if you think about how this shape of red blood cell changes, the gene, the DNA sequence is just one base change, okay? That will change in 1 amino acid change, okay? And if you have many amino acid, 1 protein, right? That particular amino acid is critical that shape the protein, okay? The shape of protein changes, and that will change the red blood cell, okay? So it's quite amazing thing, okay? But some genetic disorders are referred as a dominant, which means if you have only 1 copy of it, it will show up, okay? Good example is Huntington's disease. This is sickle cell anemia, okay? In this case, in order to have this disease show up, you have to have children between 2 carriers. In other words, both mother and father carries this allele, okay? And both affected allele meet together and become homozygous. Then they will show up, okay? So 25% of their children have chance to show this genetic disorder. On the other hand, dominant genetic disorders, if you have only 1 copy of it, okay, in this case shown in uppercase, large, the capital H, okay, this case, this person is a heterozygote and it will show up. The only problem of this Huntington's disease, they will not show up until early 40s, mid 40s, and he didn't know he had this disease. Okay, and he get married, he have children. Now, around 40, he showed up symptoms. Okay, so it's a little bit difficult to pre-diagnose this Huntington's disease. But nevertheless, he carries these alleles, and then it will pass on to next generation. In this case, half of their children will have chance to become Huntington's disease. Okay?
Some of the diseases and afflictions that affect human beings have a genetic origin. These problems may arise either from the presence of defective alleles or from various chromosomal mutations. The former category includes, but is not restricted to, disorders that can result from defects in alleles on sex chromosomes. An example, as we saw in the previous tutorial, is red-green color blindness. There are also disorders that are due to defects in alleles of non-sex or autosomal chromosomes. Some of these are autosomal recessive. In this case, an individual must possess 2 recessive non-functional alleles of a gene in order for the disorder to be expressed. Others are autosomal dominant. In this case, the presence of a single faulty allele on a non-sex chromosome can cause the disorder, even if the individual also has a second normal allele for the gene. In this tutorial, we will first consider examples of both autosomal recessive and autosomal dominant disorders. Then we will explore how various chromosomal defects can arise. Sickle cell anemia is a recessive autosomal disorder. Both the mother and father must carry at least one allele for the trait in order for son or daughter to be a sickle cell victim. When this is the case, there is a 25% chance that any given offspring will inherit the condition. On the other hand, if only a single parent has a Huntington allele, H, there is a 50% chance that a son or daughter will inherit the condition known as Huntington disease. Since Huntington disease is autosomal dominant, only a single faulty allele, uppercase H, is sufficient to cause the disease in offspring. Almost everyone is familiar with family trees, diagrams that illustrate how different members of a family are related to one another. A pedigree is a kind of family tree of a medical condition. Not only does it show how family members are related, but it also includes additional information to show the history of the condition over some number of generations within the family. A pedigree illustrates which members of a family had and which did not have the condition in question. It can be used to determine whether the condition is dominant or recessive. Sometimes a pedigree can also show which persons in the diagram are heterozygous for a recessive condition. These persons are known as carriers for the condition. They carry the allele for the condition and can pass it on to the next generation, but do not themselves display symptoms. Pedigrees are useful for couples who are thinking of having a child when there is a history of the disorder occurring in their families. Once it is known whether the condition is dominant or recessive and which family members are carriers for the condition, it is possible to get an indication of the probability for the condition being inherited in the future. It is conventional to use a standard set of symbols in pedigrees. These are explained below the figure at the left. This figure illustrates a hypothetical example pedigree through 3 generations. The Roman numerals at the very left of the figure number these generations. From the figure, it is immediately obvious that 2 individuals in the pedigree had the condition in question. But what else can be surmised from the diagram by using this starting information and the application of a bit of Mendelian genetics? We can see right away that the disorder must be recessive, since the female in generation 2 has the disorder, but neither of her parents does. Also, since the daughter has the recessive disorder, but neither parent does, each parent must be heterozygous for the condition. Apply similar reasoning to see how much of the rest of the pedigree makes sense to you. Do you understand why some family members are labeled with question marks? The condition called aneuploidy is one in which an organism has either more or fewer chromosomes than normally exist in a full set for that organism. A common cause of human aneuploidy is non-disjunction, which is a failure of homologous chromosomes to separate during meiosis. Then, some daughter cells can end up with one chromosome too many, and others with one too few. Aberrations can occur within a given chromosome, sometimes resulting from interaction between chromosomes. A frequent cause of such change is that pieces of chromosomes can break off from the main chromosomal body. A chromosomal deletion occurs when a chromosome fragment breaks off and then does not rejoin any chromosome. When a chromosome fragment rejoins the chromosome it came from, it may do so with its orientation flipped, so that the fragment's chemical sequence is out of order. This is called an inversion. A translocation can occur when two chromosomes that are not homologous exchange pieces, leaving both with improper gene sequences. If two homologous chromosomes exchanged unequal pieces of themselves in crossing over, duplication can take place. One chromosome would lose genetic material while the other would gain it. In the example shown, one chromosome experiences a duplication of the D gene while the other chromosome experiences a deletion of this gene.
Okay, so that summarized most of the rest of the part of this chapter. Okay, so we'll go through one by one. So tracking trait with pedigrees. Okay, it's a family history in genetic terms. Okay, pedigree. So pedigree will allow expert to make deduction about the genetic makeup of several generations of family member. The good example, well, in this case, the circle is female and square is a male. Okay, this is parental stage, second generation and third generation. Okay, and this person is affected that we know. So, and to determine whether this is recessive or dominant, okay, one good criteria is, the question is whether all the generations show this genetic disorder. That's one way. If genetic disorder is showing every generation, it could be dominant. But in this case, the first generation, nobody showed this genetic disorder, okay, except the second generation daughter. Okay, so we assume this could be a recessive genetic disorder, okay, and so on. Okay, in this case, this is the phenotype of albino. Okay, it's also recessive disorder. If you look at the textbook page, I don't know the page, but table 12.1, it should be same table, right? So it's a table showing X-linked disorder, autosomal recessive disorder, autosomal dominant disorder, and so on. Okay, so you can go through those tables. Okay, so next one, it didn't explain, but it's called aberration in chromosomal sets. In other words, before we go into numbers, we can have two sets of chromosome, right? That's why we call it diploid or diploid. One set from mother, one set from father, right? But you can have more than two sets, right? It's called polyploidy. Okay, so human chromosome is 46. Okay, 22 pairs, number 1 to number 22 from mother, number 1 to number 22 from father, and XX or XY, the total 46. Okay, so it's two sets. Okay, but if you have more than two copies, in animal case, it's mostly lethal. They don't survive. Okay, so you have to have only two copies. But in some plants, you can have more than two copies. It's called polyploidy. Polyploid. You can have 3 sets. You can have 4 sets. What happens if you have more than 2 sets? The fruits getting bigger. Okay, so plants get bigger. So there are some advantages in plants. Okay, that can have polyploidy. Okay, so artificially they can make polyploid, and then you have bigger fruits. Okay, so in that case, plant can survive, but not the animal. Okay. Second case is it's called aneuploidy. Okay, aneuploidy means they can have different number of it. Okay, not the whole set, but within one set, just one more or one less chromosome. Okay, it's called aneuploidy. Okay, how this thing happen? Okay, aneuploid definition. Okay, there's a condition in which an organism has either one more or fewer chromosome than the normal existing full set. Okay, so in most cases in human, okay, if you have this aneuploidy, okay, so the result will be miscarriage, which is 자연 유산. Okay, during pregnancy somehow the fetus cannot survive. Okay, except one case, which is except one case called Down syndrome. Okay, Down syndrome, a small portion of embryos survive aneuploidy. Okay, but the children who result from this embryo are born with such conditions called Down syndrome. Okay, then how this thing aneuploidy happen? Okay, we talked about the segregation of chromosome. Okay, if it is normal, it's perfect segregation. But sometimes they will make mistakes. Okay, so instead of separation, they fail to separate and then go one side, and then you will have one more chromosome in that case. In this case, you will have one less chromosome. It's called aneuploidy. Okay, one such example is it's called the thing this happening is called non-disjunction. In other words, they failed to separate. Okay, the word is non-disjunction. Okay, you can imagine all different kinds of examples, but okay. This one. Okay, so if you look at the textbook, going through page 219 in 5th edition, who has 4th edition didn't bring books. Okay, so lessons from a royal pedigree. Can you follow on the book? No, not there. We talked about. We'll skip. Okay, so if you look at the page 221. I don't have it on the slide, do I? No, I don't have it on slide. Okay. So page 221, it's about the Down syndrome. It's about the Down syndrome. Okay. The definition of Down syndrome is if you have one more copy of chromosome 21. Okay, it's very small chromosome because big chromosome is number 1, all the way. The smallest one is number 22. Okay, it's the second smallest one, number 21. Okay, if you have 3 copies of it, then it will show as Down syndrome. Okay, you don't have the whole chromosome, but if you have some small fragment of chromosome 21, it will show Down syndrome. Down syndrome patient, you probably can recognize. Okay, a little shorter statue, and face has very unique feature, and they don't live very long. The expected life expectancy is around 40, and they have some problem with heart development. They have lower intellectual abilities. Okay, so for the Down syndrome, there is no family history. It's not genetically transmitted disorder. Okay, there is no relation with, family history. The only correlated factor is the mother's age, not the father's age. Okay, that's a little bit peculiar. Okay, so if you look at the table, figure 12.8 in your textbook, Okay, it shows how many Down syndrome child at birth. Okay, if the mother's age is around 20, okay, and one case out of 2,000 case. Okay, but if the mother's age goes 30, okay, then one case out of 885, 1,000. Okay, if you go to 40, the mother's age is around 40, one case in out of 110. Okay, so the mother's age is very, very important for Down syndrome case. So it's the only thing correlates with this one. Okay, strangely, not the father's age. Okay, so how do we understand this one? Do you remember right? Do you remember how the males will produce sperm? Okay, so the spermatogonium, okay, the original cell divide once, and then once divided, this one remain the character of original cell. We call it stem cell, and then keep it as a bank deposit. And you will just take out the interest out of it, and then separate that cell will become sperm. Okay, and next division, you have one cell remain as a stem cell, and then it keep dividing until the male dies. Okay, so they'll make sperm every day fresh. Okay, on the other hand, female case, what happened? They have fixed number of egg. Okay, and then that determine, and then once a week you take out the primary oocyte and go through the meiosis. Okay, meiosis complete when this oocyte will meet sperm. If they don't meet, they don't finish meiosis. Even they don't finish meiosis, and then. So in other words, your egg, the oocyte, is aging with your age in female case. That's why if you go getting old, the separation of meiosis, the last meiosis, is not as accurate as it used to be. Okay, that's the only possible explanation. There is no proof it, but that's the correlation for Down syndrome case. Okay. Aneuploid and cancer. Okay, we talked about this aneuploid, and it's all talking about meiosis. Okay, why? Because it's preparing for the next generation. But what about our body cell? We call it somatic cell. Somatic cell will go through division called mitosis. Okay, the aim of mitosis is generating exactly same cell. Okay, but sometimes this mitosis can go wrong too in some cells in our body. If they fail to separate, what happens is they can have, a few more, one more chromosome and here and there. Then what happens is that cell become cancerous. Okay, they keep dividing and it become cancer. Okay, but this one, it doesn't go to the next generation because the cell is different. Somatic cell is for ourselves, and germ cell, sperm or egg, is for the next generation. Okay? So structural aberration of chromosome. This one is just easy. Just understand what's going on. Deletion, chromosome, some part is deleted. Probably they don't survive, okay, in this case, in human case. Okay? Inversion, some part of chromosome is flipped. Okay? Translocation, some part is located in some other place. And duplication, okay, same repeat somehow. Okay? Just remember there are 4 different types: deletion, inversion, duplication, translocation. That's all. The big question was how you can copy the genetic information exactly same way. Okay? So there was a big puzzle. Okay? But early 1940s and 50s, people were trying to find what is the structure of DNA? Okay, at that point, DNA was the genetic molecule, not the RNA, not the protein. Okay, they knew DNA is the molecule, but they don't know how they look like. Okay, they wanted to know about the structure of DNA. Okay, the form and function. Because if you know the structure, you can understand how DNA can be copy itself, replicate itself. Okay, so try to understand this structure. Okay, people were racing each other. Who's finding this structure first? Okay, the book called probably written by James Watson. It's called DNA Double Helix, 이중 나선의 구조. That was translated in Korean, so I read it when I was at high school. That's how I decided to become biologist. It was a long time ago, but anyway. So what happened was Watson and Crick, two people kind of come up with a model. Okay, this is DNA look like. Okay, and that was correct. And then they become receiving Nobel Prize. It's called double helix. Okay. Watson and Crick. Watson, James Watson was American. He finished his school very early, so he received his PhD around 25. So probably he graduated college around 20. Okay, so in the United States, it's possible you can skip some, grade, and then you graduate college around university around 20, and you receive your PhD around 25, and then he went to England, okay, for his postdoc. Crick was a physics majoring graduate student, a little bit old graduate student. He was a little bit 30 sometime. They paired up. There was good, pair. They discuss and they talk about and they build a model. Okay, so that structure of DNA, it's kind of opening up new field in biology. It's called molecular biology. In other words, the biological phenomenon can be explained by in terms of molecules, DNAs, and your bases, and things that you cannot see on your naked eyes. Okay, it's called molecular biology. Okay, these two goofy-looking guys are Watson and Crick. Who's Watson and who's Crick? This one is Watson, and this is Crick. Okay, and they built their DNA model. Okay, double helix model, and they published paper 1953. They were probably late 20s and early 30s. Okay, 9 years later, they received Nobel Prize. But they didn't do any experiment, okay? They talked about other people and other people's data they thought about, and then they built up the model. Person who did actually experiment her name is Rosalind Franklin, okay? She used a technique called X-ray diffraction, X선 회절법, okay? So how does it work? I don't know exactly, but I can tell you in an easy way. So if you have lots of DNA in solution, okay, they can form a crystal. Think about salt crystal. If you have salt water, okay, if you increase the salt concentration, there is a precipitation of salt and crystal. Okay, you get this DNA crystal. Okay, and then you put it here, and you hit it with X-ray, just like exposing yourself to the X-ray, right? Then you see the bones, with broken bones or things like that. Same thing, you hit X-ray with this DNA crystal, and then study their image on the backside. It's just like, X-ray of the DNA. And the pattern was this one, okay? And she was trying to figure out why this kind of pattern. And these two kind of look at her data, and then they, come up with this helix structure. Okay, so 4 people published at the same time. If you look at the old version, a new version. There's one more person. Okay, James Watson and Crick and Rosalind Franklin. And then there's Maurice Wilkins was another person who was doing experiment. Okay, so 9 years later, 3 people received Nobel Prize: Watson and Crick and Maurice Willis, except Rosalind Franklin. What happened to her? She died like 2 years before the announcement of Nobel Prize. Probably if she lived, don't know whether she got received or not, because she was working with X-ray. At that time, X-ray, they didn't know X-ray was harmful to human. So probably she was exposed to much X-ray. Okay, she died of cancer. All right. So now we understand the structure. DNA structure is what? Double helix. How does it look like? Okay, so we have to know a little bit of thing. DNA consists of nucleotides. In other words, building block of DNA is nucleotide. It's called nucleotide. What is the building block of protein? Amino acid, right. So building block of DNA and RNA is nucleotides. Okay, so nucleotide consists of 3 components. What is it? Base, sugar, phosphate. Okay, base, sugar, phosphate. Okay, base in DNA there are 4 different types: A, G, T, C. Adenine, guanine, thymine, cytosine. 4 different kinds. Okay, and phosphate is just phosphate. Okay, sugar. In this case, DNA it's called deoxyribose. In RNA case, it's just ribose. Okay, so if you look at the structure, what are we missing? Okay, so sugar and phosphate, they form this handrail again, this helix, and then base is in the middle, stacking. Okay, so we know the base pairing rules. A always pair with T, G always pair with C. And AT has 2 hydrogen bonds, GC has 3 hydrogen bonds. Okay, so they look like this. Okay, so this is sugar, deoxyribose. How many carbons this ribose has? 5, 6? How many carbons? 1, 2, 3, 4, 5. So it has 5 carbons. It's called pentose, means it has 5 carbons. Okay, this is deoxyribose. Okay, exactly same, but just a little different. It's called ribose. Okay, the ribose is exactly same except this one, except this H, it has OH. Okay, that's why this is deoxy. They don't have oxygen here. The other one has OH. It's called ribose. That is used in RNA. This one is used in DNA. That's the one difference. But DNA, RNA structure is totally different. Okay, and there's 4 base: A, T, G, C. You see the difference? A and G is a little bigger. They have 2 rings, right? T and C is a little smaller. Okay, that's why A always pairs with T, because they don't have enough space if it is too big. Okay, they have. So usually A means pairs with a T here by 2 hydrogen bond, and then G and C has 3 hydrogen bonds. Okay. So nucleotides. Okay, this is one nucleotide. Okay, and double helix is going this way, and one goes this way. Okay. So do we understand the structure of DNA? Okay, as soon as they propose this structure of DNA, it is clear how they can replicate themselves. In other words, how you can copy exact genetic information. We saw the movie DNA replication, right? If you open up this helix, each strand can be a template, and then you can just put the other strand again. Okay. All right, so DNA is copied by means of each strand of DNA serving as a template. Okay, template means 주형. You can put, based on their template. Okay, and for synthesis of new complementary strand. Complementary means it's not exactly the same, but if it is A, it automatically comes T. If it is G, it comes a C. Okay, there's nothing you have to, think about it. Okay. So and so. Okay. So this is how you can think the DNA replication. Okay. First, you have double helix. Okay. First thing, as we saw in the movie, you have to open up. Okay. In bacteria, it's called replication fork, replication bubble, whatever you call it. It opens up. Okay. And each strand can be as a template. Okay. And based on this sequence, you just put one by one. Okay. But there's two different. The strand, okay, is different. Why? This is double helix, right? So one strand goes here and one strand here. Okay. Double helix, we call it anti-parallel. They are parallel, but they put it anti. What does that mean? Their direction is different. Okay. Why there's a direction? Because when you put a nucleotide, okay, there's only one way you can put here, and in the others you cannot put up here. You have to put it in the other end. There is a way to growing the strand. Okay, it says 5' to 3'. So what this means? Anybody? What is 5' and 3'? Chemistry? What this number means? Great, good. Somebody answered. Okay. So we said we had this deoxyribose as 5 carbon. Okay. It's the carbon position. Okay. 1, 2, 3, 4, 5. Why did it put it hyphen? You have also carbon in base. Okay, so you want to say it's different. This prime means position of carbon at ribose, deoxyribose base, sugar. Okay, 5 to 3' means this is 5 position. This is 3rd position. Okay, it can only grow from 5 to 3 position. So if you have at the end here, okay, 5 position is occupied by phosphate. Okay, so you can put next base here, but this one cannot go in this way. Okay, so there is a direction. There's growing direction of it. Okay, so if you don't understand, it's just it has a direction of it. Okay, it's called anti-parallel. Okay. So that causes some problem, okay, when you replicate. We saw the movie, right? They say leading strand versus lagging strand. Okay? So what it means is you open up this, it's not in the textbook, so it's a little bit extra. Okay? So you opened up this one. Okay? So you want to make new strand. So you want to make red one. Okay. So new strand can go this way. You can make this way. Okay. And you can make new strand this way. So it's anti-parallel. That's fine. But once you open up from one side, what happens is, let's say you start from here. You open up, okay, this one can go this way. As this one opens, it just continuously can go. Okay? So it's one called leading strand. Okay? But what happened is, 잘못 썼구나. 여기다 쓰면 안 되는 건데. This one opens up, okay? What happened is you cannot start from here because this will opens up from here like this, okay? You have to wait until all of them are opened up and then you can start from here, but that's not the way it goes. It opens up and you start from here this way, and here this way, here this way, because of they opens up from here like this, okay? The next one, you go like this. It's called lagging strand. Lagging means they fall behind, they're slow. Okay? This one just opens, it just goes straight, one strand. Okay? This one is, you cannot do it because you open it, this is the beginning, and this is the beginning, this is the beginning. Okay? So this one is called lagging strand. Are you there? Or all lost? Okay. So that's it. We can go into detail, but it's more complex. There's two, because why this thing happened? Because there is a directionality. It always, DNA is synthesized from 5' to 3'. Okay? So it's, you can put only one side, not both directions. Okay, so leading strand has no problem. It just go straight. Okay, lagging strand. We saw the movie. What happened? There's a little red one was in the movie, right? What is it called? It's called RNA. RNA, right? You start with RNA, and then later you change into DNA, right? So what do you call it? It's called. You cannot start DNA replication de novo. De novo means from nothing. You have to have RNA primer to start DNA replication. What about the leading strand? They do have RNA. Just one. You need one. Go at the end, you can get rid of it. But lagging strand, you need more because you have to piece by piece. Okay? What is this piece called? Anybody? Yes, Japanese guy called Okazaki is the person who first discovered this one. It's called Okazaki fragment. Okay? All right. So this is how they can replicate, but all the concept is like this. You start with double helix, you opens up, and you replicate. Okay? The direction is different, so one can go from here to here straight, and the other is fragment by fragment, but that doesn't matter. But okay, you can replicate. And at the end, what you have two exact copies of DNA double helix. Okay? That's how you do replicate. Okay? So we saw in the movie there is a big, bulky thing sitting on the DNA and then doing this. It's called enzyme. What is the name of enzyme? It's called DNA, what? DNA polymerase. Polymerase. ASE, it ends with ASE. It's an enzyme, right? What it does, it can replicate DNA, right? So it's called DNA polymerase. Polymerase means small, from a building block, it will make a bigger one. Okay? DNA polymerase to replicate the DNA. Okay? And then there was one more enzyme name came from the movie. What was it? At the end you get rid of this one, right? And then you put DNA in it. But it nearly cannot fill the gap. What it is called? DNA It's an enzyme finished with A's. What is it? Right, it's called ligase. It's like glue. You put two things together. Okay? All right. So in this case, this picture, so originally it's all colored in red, right? The new strand is colored white. Okay? So at the end, one strand is old one strand is new. So sometimes we call it semi-conservative replication, which means one old strand, one new strand. Okay. All right.
The components of DNA are shown on the left. There are three groups: phosphates, nitrogenous bases, and the sugar deoxyribose. Notice that there are four common bases. Their names are adenine, A, guanine, G, thymine, T, and cytosine, C. Together, they are called base pairs, and they pair via hydrogen bonds. A always pairs with T, and G always binds with C. The building blocks of DNA are units called nucleotides, each of which is composed of one phosphate group, one sugar, and one of the four bases. On the left is an illustration of the DNA double helix. The abbreviations S and P denote the sugar and phosphate groups that make up the handrails of the double helix. The base pairs extend from one handrail to the other, forming a staircase. The complete collection of an organism's genetic information is called its genome. In each of our cells is a complete copy of the human genome. When a cell divides, another copy must be provided so that each of the daughter cells has one. The genetic information can be passed on by the process called replication. During replication, the two strands of DNA that form the double helix separate, serving as two template strands for replication. Free nucleotides base pair with their complementary nucleotides on the existing template strands. The result is two identical strands of DNA. Each replication process combines an old strand with new nucleotides. This model of replication is called semi-conservative replication.
DNA replication has to be very, very precise. Otherwise, you cannot replicate, you cannot copy exact genetic information, right? So DNA polymerase. So let's say, what is the size of human genome? Anybody knows? How many bases, how many base pairs? Human genome? 30억? 3 billion, exactly. 3 billion nucleotide. So let's say DNA polymerase, the enzyme, makes mistake once in million. Then how many mistakes will it have? Million? 1 in million? Billion? You have thousands of the mistakes, right? So you cannot have that mistakes, right? So DNA polymerase has this editing property. In other words, we call it proofreading. Proofreading is you write something and you read again and you change, okay? So DNA polymerase puts nucleotide, but then if he makes mistakes, he go back and corrects it. Okay, that's what it's very, very accurate. Okay, so they can probably 10 to minus 9th. So almost, if you replicate the whole genome, maybe they'll make 10 mistakes or something like that. All right, it's very precise. Okay, but they'll make mistakes. What happens if they make mutation? They make a mistake. What happens is, let's say the original information was GC. But I make mistake. You have to put it C, but instead of you put it T. Then what happened? Next generation, it will change one strand. At least one strand will change into A and T, right? So genetic information is changed. It's called point mutation. Okay, so it may be important or it may not be important, but usually mutation is negative. In other words, the original information is changed, then it will affect in a negative way. Okay? But if you, let's say, in somatic cell, in your body cell, make mistakes, the worst case is you get cancer, right? But if you have mutation in the germ cell, in other words, your oocyte or your sperm, that mutation goes to next generation, right? There's a difference where the mutation occurs, germ cell or somatic cells, okay? And if you make have a mutation in germ cell, it's called heritable, in other words, next generation. But in somatic cells, no, it will not go to next generation. Okay? So in other words, it's called mutagen. It's a substance that cause this mutation. It's called mutagen. Okay? So examples, what? Cigarette smokes or the UV lights. So UV lights can change your DNA base in your skin. So sometimes skin cancer, okay, is caused by this UV. Okay? But the other side of mutation is the only source of change in DNA information, genetic information. So if you think about long run, like evolution, okay, the mutation is the driving force that changed genetic information. Okay, we'll get back to it when we go to the later chapters, evolution. Okay, it's very, very rare instances. Okay, but it's sometimes, do the trick.
When a cell's DNA replicates, mistakes sometimes occur. Fortunately, there are a variety of mechanisms that check for and repair many of these mistakes. Consequently, the error rate at the end of replication is very low, but it's not zero. When a mistake in replication is not corrected, the result is a mutation, a permanent alteration in a cell's DNA-based sequences. Many mutations have no effects. When there are effects, they are most often negative. For example, cancers can result from mutations that cause cells to reproduce uncontrollably. But mutations are not always harmful. In fact, they are the only way entirely new genetic information arises in an organism's genome. When mutations occur in either egg or sperm cells, the changes can be passed from one generation to the next. Mutations thus play a vital role in evolution. A point mutation is a replication error that occurs at a single location in an organism's DNA. One example is that a slight change in the chemical form of a base on one DNA strand can cause it to link to a different base than it otherwise would. For example, a mutation might cause a G to link up across the helix with a T instead of a C. In another example, a sequence of base pairs along the handrails of the DNA double helix might be inserted in error.
So that's about it for chapter 13. And so I uploaded the crossword for chapter 13 this week. Okay, so by next week, you have to hand in chapter 11, 12, 13. Okay, so next week we'll go through chapter 14 and 15. Okay, any questions? Okay, so I'll see you next week.